Hi, everyone, and thank you for joining us today. Welcome to today's Tips and Tricks webinar focusing on extracting meaningful data from super resolution images. Before the presentation begins, I'd like to go over some quick house rules for the webinar. First, the presentation will last about 45 to 50 minutes, leaving approximately 10 minutes for Q&A at the end. You're welcome to submit any questions you have throughout the webinar via the chat section located within your panel. We'll be answering your questions during the Q&A session. If we're unable to get to your questions, we'll email you your answers separately via the email address that you registered with. So within your attendees panel, you're going to find a handout section. And here is a company brochure about ONI that we'd like to share with you. So please feel free to have a look there. Please note that today's webinar is being recorded. It will be stored on the media section of our website for future reference, but if you'd like a copy of the webinar as well, we'll be sending everyone a thank you for attending email containing a video copy of this webinar. And now I'd like to introduce you all to our speakers for today, Dr. Corey Butler and Dr. Marcus Fantham. Starting with Corey earned his BSc in biomedical engineering from Duke University, followed by an MS in optics from the University of Central Florida and a PhD in bioimaging from the University of Bordeaux. During his time as a PhD student and postdoctoral researcher in Bordeaux, Corey integrated advanced microscopy hardware with specialized software to push the limits of single molecule super resolution microscopy. Corey is currently the product manager for CODI, which is ONI's collaborative discovery software, ensuring the CODI team is developing solutions that deliver high value to customers in an easy-to-use package. Now on to Marcus. Marcus earned his master's in engineering from Oxford University and continued his studies at the University of Cambridge, where he obtained his PhD. During his PhD research, Marcus focused on improving the SIM technique for super resolution microscopy. His work involved developing and refining algorithms for faster reconstruction times and analyzing large amounts of data captured from biological experiments. His contributions have advanced the field and have been recognized by his peers. Currently, Marcus works as a software engineer at ONI, where he applies his expertise to develop cutting-edge software solutions for scientific instruments. His work has included designing user-friendly interfaces and developing software for instruments used in a wide range of fields. Okay, and with that, I will now hand over the presentation to Corey and Marcus to begin. So over to you both.
Thanks, Carolyn, for the introductions. Very nice. So thanks everyone for being here today for this really exciting webinar where we'll be talking about extracting meaningful data from super resolution images from our single molecule systems. So just kind of getting started, what we're going to talk about today, I'll go through a quick introduction of how we generate single molecule super resolution images and why quantitative imaging is important. We'll get into how we can analyze some of the single molecule data with different kinds of algorithms. Then Marcus will walk us through a live example on CODI, which is our cloud-based online discovery platform for single molecule analysis. Then we'll run through some examples of quantitative analysis that we can do after processing on CODI. And then we'll go through some questions and answers with you guys. So before getting started, we've got a quick question for the audience to see if you guys are awake and listening. An easy one though. Which of the following is not a super resolution single molecule localization microscopy technique? So in this list of 5 acronyms, we love our acronyms in single molecule localization microscopy, right? One of them is not a single molecule localization microscopy technique. So I'll give you guys maybe 30 seconds or so to respond here and see what you guys think. This is a little bit of a tricky one, not gonna lie.
All right, and right now, Corey and Marcus, we have 30% of people voted, so I'll just wait a few more seconds for it to at least get up to maybe 60%. All
Looks
right.
like some answers are coming in. Some of them are obvious, some of them aren't so much. All right, great. This is what I was really hoping for. So actually, there was a really recent paper called TARDIS. It came out from Ulrika Endesfelder's group. It's on bioRxiv. You guys can, look on bioRxiv for this. It's kind of an SBT palm technique that's come out. So I thought this was a fun one. If you guys are curious, just go ahead and Google that, and you'll be able to see some. Yeah, there we go. Some new research is coming out of her group there about TARDIS. So the one that wasn't a single molecule technique was actually SOAR. So congrats to the 19% of people who saw that. All right, well, just jumping right into it now. Why is quantitative imaging important? So if you're here, again, this is a tips and tricks webinar. So just want to kind of at a very high level help make sure that everyone understands that single molecule localization microscopy isn't just the beautiful pictures with the very high resolution that we can sometimes see. Quantitative imaging of single molecules allows us to put numbers behind molecular properties like spatial organization and dynamics. It also allows us to, build a lot of confidence in our statistical power with technical replicates, experimental replicates, and being able to really compare quantitatively different experiments. And then eventually draw biological conclusions from those quantitative comparisons that we were able to draw. So on the right here, you see an example of a quantitative image of an extracellular vesicle. We get information about the size, but we also get information about the number of localizations for each of the 3 different tetraspan and biomarkers that we have labeled here. But to understand quantitative imaging for single molecule localization microscopy, it's really, really important to understand how we generate our images and how we generate our data in localization microscopy. So I just want to run through kind of some of the basics so that we all understand exactly what's happening. So what we do is we have a labeled molecule. So this is a fluorescent molecule that will be attached to a protein or another biomolecule of interest, usually by an antibody or something like that. When we're imaging it, we send some laser excitation, right? And then we receive fluorophore emission that is emitted in all directions. That's my hand. Okay. What's really interesting though is that these individual labeled molecules, right, these individual fluorophores are only a couple nanometers in size. But what we see on the camera when we're imaging them through a microscope is a large, what we call point spread function that is a couple hundred nanometers in size, right? And so the resolution of our microscope is actually limited by this diffraction limit of light, where we can't actually see the tiny molecules that we're labeling. We see these large hundreds of nanometer point spread functions. But in single molecule localization microscopy and other super resolution techniques, we play different kinds of tricks to be able to get around this diffraction barrier. And so in single molecule localization microscopy, we make the very strong assumption that there's only one single molecule that's underlying this point spread function on our camera. And then we can use a little bit of physics to approximate the localization or the location of the underlying fluorophore with a precision that's about 10 times better than the resolution of the image that we received on the camera. So this is a tips and tricks webinar. Tip number 1 is this in single molecule localization, it's really, really important that you're in single molecule conditions for the localization algorithms to work correctly. So then how do we generate a super resolution image from this process? So if we're trying to image an underlying structure, here we've got, for example, like a kind of ring structure. If we look on a wide field image, we see everything that's quite blurry and it's quite difficult to understand what's going on there. There's different kinds of single molecule techniques, and we have different acronyms for the different ways that we enable the single molecule imaging. The one I'm going to explain here is kind of the most common one, where it's called DStorm, where essentially what we do is we put most of the molecules in a dark state. So when we send our laser excitation, we don't actually receive their fluorescence on the camera. And by doing this, we're able to enable stochastically or randomly just one single fluorophore or one single fluorescent molecule on the structure of our surface at a single time. So then we can go through that localization process and pinpoint its location, again, with a resolution of a couple tens of nanometers. In single molecule localization microscopy, we repeat this process for hundreds or thousands or even tens of thousands of imaging frames to be able to receive the fluorescence and then do the localization process of each of the fluorescent molecules that's used to tag our underlying structure. And then what we generate at the end is not necessarily an image, but it's a point cloud that contains all the locations of the different localizations for the underlying emitters. So again, tip number 2 is sometimes we do have images that come out of our super resolution systems, but the data that we're actually generating in super resolution is a point cloud. It's location data. It's basically localization data of where the location of the underlying fluorophores are. So now that we understand the process and how we generate super resolution images, we can talk a little bit about how we can analyze the single molecule data, which in some ways are similar to what you can do in conventional image analysis, and in other ways is a little bit different. So there's lots of different algorithms and ways that we can extract data from single molecule imaging depending on what we want to do. I'm going to run through a few of them here very quickly, but you can refer to our website. Anna from our team at ONI here just wrote a really great article about how to analyze data using single molecule imaging. So please check out our blog to learn more about that. There's a lot more detail than what I'll be able to cover here. But some of the primary algorithms that we use are clustering. I'll run into that one a little bit more in detail in a little bit to explain a little bit better. But the idea is that in our localization data, we would like to be able to understand where different aggregates of proteins are, or different aggregates of localizations to help understand what the biological implication of that localization data is. We can also run segmentation. Segmentation would allow us, for example, to say, okay, in this particular part of the cell, I might be looking at a specific protein or a specific organization of a protein in a different part of the cell, in a different segment of the cell. It's also really common to run things like near neighbor analysis to understand the heterogeneity or the homogeneity of the distribution of these localizations across your cell or across your field of view. It's also really important to note, like in fluorescence, right, we've got this awesome power to see individual things that we're tagging, right? So individual biomolecules that we're tagging with fluorescent labels, and we can do that with multiple colors, right? So co-localization is a kind of standard technique that's used in conventional microscopy that we've kind of ported over also to single molecule localization microscopy that allows us to say, to give us information about the co-organization of proteins or other biomolecules. And then I think it's really interesting to talk about some of the other in the past, 10 years or so, single molecule specific methods, for example, to estimate the image resolution by calculating the nearest neighbor, not spatially, but actually in the adjacent frame. So it can give us information about, again, what is kind of the precision of the localization that we're able to do from that point spread function, and give us a good indication of the resolution of our image at the end. So again, just running very quickly through a bunch of these different methods. Today we're going to focus on clustering because it's kind of, if you think of the single molecule localization microscopy analysis toolkit, clustering is the first one. Clustering is your hammer. It's the first thing that you're going to use to go in there and say, okay, well, what can I see in my cells here? What can I see on my fluorescent tags? Clustering is going to be the tool that's going to enable us to see different receptors clustering, for example, or just to identify individual fluorophores on the cover slip, for example. So just a very brief introduction to clustering. The idea of clustering on localization data here is, if you look at these three sets of green points, you can imagine lots of different ways of understanding what these are. It could be one big aggregate of localizations that is one big tag, or it could be a bunch of smaller individual clusters within there. And so this idea of clustering in super resolution is really, really important because it's going to help us identify if there's potentially, again, a single fluorophore, maybe potentially stuck to the cover slip that we're not interested in looking at, or if we're looking at larger clusters with more localizations that are probably more relevant for the specific receptors that we're trying to look at, or the different clusters of receptors. So just as a very high-level overview, here's an example showing you the clustering algorithm working. So on the left, we've got the conventional D-storm image of some extracellular vesicles stuck to a cover slip surface that have been captured on the surface, where the color code is just the localization data. And then when we run our clustering algorithm, you can see on the right here, the color code refers to each individual cluster. So the clustering algorithm is going to actually be able to automatically separate the extracellular vesicles one from another that allows us to do a specific single extracellular vesicle analysis. All right, well, so now that we kind of understand what clustering is, I'm gonna hand it over to Marcus in just a second here to show us on CODI, our collaborative discovery platform, how we're able to do these kind of analyses. And just very quickly, CODI is our cloud-based analysis platform where you can collaborate with colleagues and collaborators by sharing not only your data, so your single molecule localization data, but also analysis workflows and analysis results from anywhere. So you can do it from the computer where you performed the acquisition, or you can do it from an analysis computer that you've got at home or in the office. It's a really great platform, and Marcus is one of the primary developers for CODI, so he's going to walk you through the CODI platform right now.
Awesome, thanks. If we come to this screen. Cool, so this is the CODI home screen. If this is your first time seeing CODI, you'll see that there's loads of beautiful data that loads in immediately. This is all running on the web, so you can access this yourself from the website. You'll have to ask us for the link. If this isn't your first time seeing CODI, then you'll see that we've got some new features, which we call apps and batch analysis. So this is going to help us really extract lots of statistical information from our data from across multiple acquisitions. If you've been running an experiment for a day, you can then analyze all of that data just in one go. And so I'm going to demonstrate some of that today on the platform. So I hope old and new CODI users can get something out of this. On the side, we have various galleries. You can see data sets that have been shared with you privately. There are data sets that people have chosen to make public, perhaps after they've published a paper. This way they can share the data, and readers of their paper can come onto CODI and either rerun the analysis themselves to verify the results or start running their own analysis on this now public data. I have a gallery of my own data sets that I've uploaded, and today I'm going to start with some nuclear pore data. So the first screen we see when we load in our acquisition, our data sets, is this sort of metadata page. We have a dynamic gallery here of any views that anyone saved on the data. So these include the analysis. So after you run some analysis, you might want to save that view down, and that will appear in this gallery here. We have ways of tagging the data. We can add a description to the data. We encourage people to add descriptions. There's a social area, so you can leave comments on other people's data sets if this is shared. At the bottom, we have any analysis that's already been run on this data set. So this data was uploaded a little while ago, so it's already had some analysis run on it. We're going to run some more today, and that will appear in this table. This is where the collaborative aspect comes in the sharing region. So I can choose to share directly with some users, or I can make the entire data set public and then just share the link. And anyone with a link will then be able to come and see the same data set and interact with it as I'm about to do. A little bit of metadata from the microscope itself to help you repeat this experiment yourself. If you want to use exactly the same conditions, the same number of frames, the same laser powers, for example. We can name our channels. Again, we encourage people to do this. I haven't done it here. And finally, we have a rating star. And so you can rate by 5 categories, and this can help you sort your data and find your best data sets if you're maybe trying to share an example for people. So if I click on the gallery, it will take me to this live preview. This is nuclear pore data, but something looks a little bit off. There's lots of streaks. And if I zoom right in, we can see each individual localization that Corey was describing earlier. So this really is a point cloud of data. And so each localization we've just placed down on this canvas. It does create a beautiful image, but remember that the underlying data is all localizations. And so bearing that in mind, we can actually start visualizing the statistics and the extra information we gather about each localization. So one of them I'm going to color by here is called frame index. And so I now have colored in blue frames from the beginning of my acquisition. And then I think this was I think it was 15,000 frames long, this acquisition. And so the 15,000th frame is colored in red. And we can see it sort of making streaky rainbows across this data. So the first thing we probably want to do is run drift correction. This streak may have come from thermal expansion, or maybe just something sliding on the cover slip. We'll see in a second just how many nanometers it slid. So I click on the Add New Analysis. And so I'm then presented with this gallery or this app store of different analysis apps I can run. At the moment, all the apps are provided by ONI, but as this platform grows, we're hoping that contributors will add their own apps and their own analysis tools to the platform. For this nuclear pore data, I can choose what sort of analysis I want. And today I really want to get some information about the morphology of each nuclear pore out, so maybe the size and the shape. And so this clustering app sounds perfect for me. It's a little description saying we're going to find statistical and morphological properties of the data. And on the right-hand side, we can see it says clustering, and it's got a list of the tools that we're going to run. So if I click on it and choose some preset settings, it will load it in. Okay, and those 5 tools that were listed have now appeared at the side, and we can step through them one by one, or we can choose to run the whole thing in one go. For now, I'm going to step through them one by one so we can discuss each step. So it's as simple as just pressing play. CODI has been designed so that you shouldn't need to know any technical information about how this algorithm runs if you don't want to. So just by hitting play, we've chosen some good parameters by default, and we can see that streaky rainbow effect after we run the drift correction actually shows us that we have some beautiful nuclear pores sitting in the nucleus of our cell. And we can see that we had maybe 700 nanometers, say, of drift in this example. If you do want to go and optimize the parameters further and you're feeling technically minded, you can press this menu button, which will reveal all the parameters that can go into this tool, into this algorithm. And if you need more information about them, you can hit the big question mark button, which will open up a how-to guide. The next step in this workflow is filtering. So again, I click on it and we can see that some filters have been pre-selected for us. Some of them have very obvious names like localization precision. Some of them like a p-value, slightly more ambiguous, but if you hover over it or go to the back and click on the question mark, again, you'll be able to read exactly the maths behind what each of these filters represents. And now we'll hit play.
Do you mind if I jump in, Marcus?
Please do.
So as this is kind of an extracting meaningful data from the super resolution images webinar, I just want to note that those 2 first steps here, the drift correction and then the filtering, are super important. They're really, really critical to making sure that we have quality data that we're going to be analyzing downstream. So here, the preset settings that Marcus is running through are what we suggest is using as kind of the typical filters that basically remove anything that we think is not a single molecule that has been localized, right? So for example, to your point, there's like the sigma. Sigma is about the size of the point spread function. And so we put some filters on here that limit the size of it. So we're filtering out the ones that are too large and probably are 2 molecules that are close together that are fluorescing at the same time. We're also limiting here, for example, the localization precision to a very highly precise, so very low numbers here, to just keep the most, like the highest precision data.
Yeah, and actually the third step of filtering, you can think of it as another filtering tool. The third step of data preparation is what we call temporal grouping. So I'm going to run this. And what temporal grouping does is remove any molecule, any localization events that appear in adjacent frames and are in the same location. So actually, I think the how-to guide here explains this with a nice diagram very well. And so we can see we have one single blinking event, but that's caused a localization event in 3 different adjacent frames. And so we'll turn this 3 localization event into, come back, a single temporal group. We have an ROI building tool, so we can see there are still 1 or 2 localizations despite all of our data cleanup falling outside, and so I can quickly just outline my cell, clicking around it. And what this region of interest tool is going to do is only run subsequent analysis within this region. So a little bit more. Beautiful. And so finally, we come to the clustering, the meat of this tool. And now I can hit play. And it will run our clustering for us in this region. And as it's running, I should point out, all of this is running in the cloud. None of this heavy lifting analysis is running on your computer. So this means you can run CODI from a tablet or from a mobile phone or from any laptop, even if you feel it's underpowered. The heavy analysis is running in the cloud.
There's also a couple different clustering algorithms. Maybe you can talk through those a little bit, Marcus.
So at the moment, we're running our in-house algorithm, which we call constrained clustering. This allows us to constrain the clusters that we're creating by various parameters on each cluster. So say I don't want to see, so in this case, the circularity parameter has already been set at 0.5. This means that I'm only going to keep clusters, or only identify clusters that fall within this circularity region. Alternatively, we have dbScan star. I think I'm going to use that on our next data set that we analyze. So we'll get to see it in a second. Here we can see all the nuclear pores have been identified individually. And so I think this clustering algorithm has been a success. If I click on each one, I can start seeing statistical information about the individual nuclear pores. And furthermore, I can see exactly where that lies on my distribution in this graph here. So I can see circularity. I was interested in the radius. And so we can see here, our mean radius seems to be, in fact, we have a whole list of statistics. Our mean radius is exactly 38 nanometers for each cluster here. And then I can click on each cluster to see exactly where it's falling on this distribution. I can now start extracting this data, maybe for a publication or a paper. I can either download the graph directly, or if I want maybe to create my own graphs in Prism or even Excel, or your favorite statistical analysis tool to then match the rest of your data in your paper, you can download all the cluster information as a CSV file. And if we open this up, we should see thousands of clusters that have been identified. And so this table just contains all that same information that we see in CODI, but in a very raw format for you to do what you like with. Okay, so this ran on one single data set, and there are thousands of nuclear pores in here, but each cell might be different. And so next, I want to show with a slightly different example some analysis run on multiple acquisitions. And so here I'm going to come to our collaboration space. And so this is a space in CODI where you can group data sets together and then share the entire group with your colleagues or collaborators. And so my colleague Chloe has already uploaded or captured and then uploaded all of this beautiful EV data. And so in this collaboration, this data is now all grouped together. We can add a description for the entire collaboration. We can share the entire collaboration with other users. We'll come back to this area in a second. I'm going to show that we can group the data. So if we've tagged our data, we can group them by that tag. Somewhat more powerfully, we can use key value pairs to organize the data. So maybe you've got different conditions in different lanes on your cover slip, or maybe the cover slip itself, or the chip as we often call it, when they've got lanes etched into them, would have different conditions. And so if we click on this, it will filter just those examples for us. Okay, we can see this particular data set has already had a specific screenshot saved on it. So if I click on it, we'll see that in the data gallery here. And here we can see there's a little bit more metadata than our previous example. So well done, Chloe. And this is the analysis that has been run previously that I want to dive into. And so I click on it, and all that previous analysis, this is all green already. We don't have to step through step by step. It's already been run. If I zoom out a little bit, we can see our clusters. So this is EV data rather than nuclear pore data now. And so each EV, each extracellular vesicle here, has 3 different fluorescent labels on it, labeling 3 different proteins. Actually, if I hide the cluster information itself, we can see underneath the 3 different proteins represented by the 3 different colors. And this one has all 3 colors on it, so we'd call this a triple positive EV. Perhaps we can find an example of a double positive EV. So this one only has yellow and magenta. So that's the CD9 and CD63 proteins. And we can see there's maybe a little bit of CD81. Now this might be noise, so we can set a threshold on what we count as a positive cluster or a negative cluster. So in this case, we've said it has to have at least 1 localization in it for it to count as positive in the CD81 channel, and then 5 localizations for both the CD63 and CD9 channel for it to count as a positive cluster. And using these conditions, we see that 55% of our clusters count as positive. This graph is very interactive, so if I want to see perhaps an example that's very positive in the CD81 channel, I can click on that and it will jump us straight to that example of that EV, and we can see indeed there's a lot of cyan in this particular cluster. Equivalently, I can click on a cluster and it will show me on the graph, or on the scatter plot here, exactly where that cluster lies. again, this is beautiful data for one example, but we saw in the collaboration, in fact, it's still open here, that I have many data sets with thousands and thousands of EVVs across the entire experiment. And so now I want to start batch analysis. So if I click on batch analysis and choose that same analysis that was run, so this was an EV profiling one, I can click EV profiling essentials. Now I can select which datasets I want to run this on. I'm just going to hit all and hit run analysis.
So this is a new feature that we just rolled out about 2 weeks ago now that allows you to use the same settings that you just set up for the first time as you're digging through your data and apply those same settings to all the different data sets that you might have in a given collaboration. So this saves a lot of clicking and saves a lot of time.
Yeah, so previously you'd have had to go through each of these data sets individually and run the analysis on them one by one and then download it one by one. What this is doing is starting up analysis on all the data sets in this collaboration with those same settings that we just saw for this individual one. This might take a little while, so here's one I prepared earlier. So firstly, we can see all those analyses that I just started up running. And so this is the analysis results section of the collaboration. Further down, we have some that I ran 7 days ago. And so if I want to extract all this information again into a CSV, batch download, I'll select the analysis results that I want in my export. Let's go to there. And then I hit download 14 results. And so now what CODI is going to do is collect all those CSVs into one single large file for us.
And again, this is all happening on the cloud, right? So you can close this window, you can go grab a coffee, go grab lunch, and come back, and your data will be here ready, waiting for you when the processing is done.
Yeah, so we can see all these analyses results still spinning. Eventually, some of them will get tick marks next to them. And I should, any second, see another download. There it is. And so this is giving me a zip file. Let's open it up. We can see it's already got some 4 CSV files inside it. And so maybe I want to see all the clusters again. This is a large file because we had that all 14 of those data sets, and every single cluster from those 14 data sets will be in this CSV file. Let's count up how many that is. 101,000 clusters. So from this, again, you can put it into your favorite statistical analysis tool and create your tables and graphs and draw whatever biological conclusions you like from them. So with that, I will hand back to Corey. We can see some of my analysis that I just ran has already finished. The rest of it's still processing. Corey,
Sure.
Where's your presentation gone?
Yeah. Great, thanks Marcus for running us through. So Marcus gave us some great examples of the different app workflows that we have by default in CODI. So the first one was just kind of standard clustering. And the second one was the EV profiling workflow, which is very specific for extracellular vesicles, where we've kind of defined the clustering algorithm, the filters, and an output of a kind of a positivity report that tells you different subpopulation information about your extracellular vesicles. So Marcus did a great job of going through each of the individual clusters, individual EVs, and showing single positivity, double positivity, triple positivity. We actually have a report that's generated that also shows you all that information that you can use to kind of analyze your data in a more quantitative way. So I just want to show you some of the kind of downstream analysis that you can do after you've done this analysis in CODI, is either with an individual data set or from the batch analysis tool. So typically, for example, we're talking a lot about extracellular vesicles today. If you are looking at your EVs, questions you might be asking yourself are, what are the different sizes of my EVs? What does the size distribution look like? And how does that vary with the biomarker? So is there a specific subpopulation according to a specific biomarker like tetraspanin, like CD81 or CD63? And is the size different for those particular ones? And so with just some statistical analysis downstream, in, again, your favorite tool like Prism or Excel or Origin, you can do some simple plotting. Here, what's really interesting in the graph in the middle here is it shows that for the single positive EVs, so those are ones with the blue lines, so it's either CD63 or CD81 positive, we have a pretty broad distribution of sizes, but relatively small. And then the double positive ones that are double positive for CD63 and CD81 are a little bit more peaky with a size around 80 nanometers. And what's really interesting is that those that are positive for the tetraspanins are quite a bit larger than the EVs that don't have any tetraspanin content on them, as you see by the yellow line. So this is just kind of a simple example of showing how some of the downstream analysis you can use to kind of quantify your subpopulations. But it's not just about EVs. Marcus also showed some other examples from nuclear pore data, and then we also have some receptor clustering here in T cells, where you might want to be investigating the efficacy of a certain kind of antibody on a receptor. And so one way of doing this is to measure in the same size, to measure the number of clusters or the number of receptors that you have in that certain area in a cell, and quantify the density of that, right? So by quantifying the density and the number of clusters in a certain area, you can start to make comparisons between different types of either antibody capture or the efficacy of different antibodies, for example. And here's just kind of my last wrap-up slide that I think does a pretty good job of showing all the power of super resolution imaging and the clustering. So here's some B cell data that's stained for CD20. If you take a wide field image, this is what you see in red in the center here. You don't really see very much of anything at the single cluster level. With the D-storm image, you're starting to be able to perceive some individual receptors, and then the clustering tool is actually able to extract the individual receptors and get you information about the distribution, the number of them, the arrangement, and again, kind of the clustering of these clusters. And you can do some kind of downstream quantification, the number of localizations per cluster, cluster size, and kind of compare these between different experimental conditions to help understand, again, how a certain antibody is working or a certain capture surface is working for your experiments. So with that, it's kind of the wrap up for the quantitative analysis webinar today. I would love to open the door to any questions that you guys might have, and hopefully Marcus and I will have some answers for you. So feel free to ask any questions.
All right. So thanks so much for the presentation, Corey and Marcus. I'm sure everyone has learned some new tips and tricks today on how to extract meaningful data from your super resolution images. And we're now going to open the floor to some questions, which Corey and Marcus will be answering live. So once again, if you have any questions, please feel free to drop them into the chat box. Looks like we have some questions submitted already. For the first question, how can I best determine whether two proteins are co-localizing within the same structure or interest?
I'll take that one, I guess. Sure. So co-localization, as I mentioned kind of earlier in the presentation, is something that's really important at the single molecule level. And when we look at them, when we look at co-localization with standard image analysis tools, it's usually like at a pixel level, what is the intensity of the two different proteins or the two different channels. It's a little bit more tricky in single molecule, but that's why this is a tips and tricks webinar. So that's the EV profiler tool that I showed you, that Marcus showed you earlier, and then I showed you some data from earlier. Essentially is looking at, for a given cluster at a certain radius around it, it's going to be able to count the different number of localizations in each channel, right? So our counting tool in our clustering algorithm works really well for that. But there's also all sorts of other methods. There's Ripley analysis, there's tessellation, there's all these sorts of analyses that you can do to kind of compare clusters in 2 different channels and see if they're very spatially close together or not. Another one is SODA, which is really great for statistical analysis and understanding the different, if 2 kinds of receptors or 2 kinds of proteins, if they're co-organizing or not. So long answer short, there's a lot of different algorithms that you can use to do it. Some of them hopefully will be showing up here in CODI to help you guys shortly.
Great, thanks so much for that, Corey. Okay, next question we have. What does it mean for the image and data if the drift calculates no drift?
Yeah, if it calculates no drift, then that should mean that you have, there has been no drifting in your during your experiments, during those 10,000 frames. You can verify this by coloring by the frame index, as I showed. And you should see no rainbow streaky patterns. If you do see some rainbows, then if you like, you can go to that advanced part of the tool and start playing around with some of the parameters. I would suspect if the default parameters haven't found any drift, then that's probably accurate and you don't have drift. But if you do want to go and play around with those parameters, then hit the big question mark and it will tell you in detail about each parameter, and you can fine tune and optimize them. And then of course, save those parameters down and use them across the rest of your analysis. And you can save your favorite parameters with the little star button, so that will save them as a favorite. And then they'll always appear at the top of the list for you to select every time. I hope that answers the question in some detail.
Great,
Feel
thanks.
free to reach out to us specifically afterwards also if you need some help, if you have a specific question.
All right, on to our next question. Is CODI available to those capturing their SMLM data using other nanoscopes other than the Nanoimager?
So I'll take that one. CODI right now is in beta and it's open to any Nanoimager customer and Nanoimager user. So the answer to your question is no, not right now. But if you are a Nanoimager user, you can use CODI as much as you want.
Okay, great. We had someone submit multiple questions, so we'll do this one question at a time. So they said, I have a few questions. First, what is the limit on data for upload in CODI?
I'll leave that one to you, Marcus.
Yeah, so at the moment, there's no limit. If we're talking about individual data sets, there's no limit on the file size. You're uploading the localization data. You can choose to upload the raw TIFF data as well for archiving purposes, maybe. CODI doesn't display that raw TIFF data, but you can download it again later. In terms of the localizations itself, there's no limit on the size, and hopefully you saw that CODI was running quite fast no matter what data set we looked at. And that's because the data is stored in the cloud, and we only present a small subset of the localizations as sort of a visual preview. If you zoom into any area in the data set, you'll see the individual localizations, and it will pull in sort of in a Google Maps style where you don't see every single road name when you're zoomed out at the country level. As you zoom into any area in your data set, the cloud will fetch more localizations for you to see. Now, when you run the analysis, because that's all running in the cloud, that analysis will run on your full data set, no matter how large it is. The larger the data set, usually the longer the analysis will take, just because there are more clusters to find or more localizations to measure the distance between if you're running clustering, for example. yeah, in terms of how many files you can upload, it's completely unlimited for our Nanoimager users, especially while this software is in beta. We really want people to upload and use the tool and not only get important data and meaning out themselves, but also provide us with some feedback for what would be the next most important tool for you to have on the platform, and yeah, any ideas for improvements or further analysis.
Great, thanks so much, Marcus. All right, a few more questions coming in. Would temporal grouping be able to provide data on time or dark time for downstream analysis in DNA paint?
Yes. I don't think we have a tool for this on CODI yet, but we have been writing some sort of in-house tools at the moment that right now, I think we just have them as Python scripts, but they will soon appear on CODI. So our sort of first stage of creating a tool is to write a Python script, and then we'll turn that Python script into a CODI tool, and then it will appear with its user interface. We can decide what sort of graphs we would like, what sort of output we want from that tool. And at the moment, we're writing these all in-house, but the vision with CODI is that anyone would be able to write their own similar Python tool, upload it to CODI, have a really simple interface for building that UI, building how we're going to show that result. And then, yeah, it will appear as one of those workflow steps. So the answer is not yet, but soon.
And please reach out to us if you have specific needs for that. Feel free to reach out to us and we can chat through some options.
All right, great. Seems to be lots of interest in the drift correction. So one person asked, what are the main differences between the drift correction methods available in CODI?
Yeah, so the algorithms themselves are all detailed in the big question mark button. I think we have 3 methods at the moment. One is called direct cross correlation. One is called redundant cross correlation. And I can't remember what the third one's called. I think I don't want to go too deep into the technical details now of how the algorithms work, but I would suggest reaching out to us, and we can put you in touch with the people that wrote each algorithm. And if the help file, the big question mark, doesn't give you enough detail, yeah, reach out to us.
All right, great. Okay, next question. In DStorm, how do you distinguish the individual molecules if there is overlap in fluorophore emission in a single frame?
Yeah, I can take that one. It's kind of what I was showing on some of the slides in the introduction. It's really, really important that we're in single molecule conditions to do all this kind of downstream quantitative analysis. But because DStorm itself it's in stochastic, right? It's in the acronym. It's random about the blinking. We can't really specifically control that there's only one fluorescent molecule in a diffraction limited spot on the camera, right? So it's kind of a guessing game, not a guessing game. It's like just a statistical random process. And that's what all those filters are really great for. So the filters that Marcus was showing us earlier on CODI that limit the size, so it's the sigma, the size of your point spread function, the number of photons also is a good indicator. You can kind of play around with your filters to say, okay, I'm only going to keep the single molecule, the localizations that are the right size and the right intensity in photon count to be a single molecule. So if there's several, so if there's 2 or 3 single molecules that are fluorescing the same spot at the same time, the spot might be a little bit larger, it might be a little bit brighter than what a normal single molecule would be, and so we can filter those out in the post-processing that Marcus was showing us earlier.
Great. All right, next customer question is, what language is CODI written in, and is the code open source?
So different parts of CODI are written in different languages. The analysis tools themselves are all in Python. So actually, some of them have been optimized with some Rust code as well, but generally Python. At the moment, it's not open source while it's still in this sort of closed beta stage. As we add customer analysis tools, yeah, we'd like to show the underlying algorithms and how they're actually working for reproducibility. Other parts of CODI, so the part that you see is web-based, so that's in JavaScript. And then the cloud itself runs in a language called Go, and that ensures robustness and means that CODI won't crash when hundreds or thousands of people log on all at the same time. I suspect those latter two are maybe a bit more detailed than you'd like, but the Python is the main analysis language.
Okay, great. Thanks for that, Marcus. Next question. Is there a way to assess how good someone's analysis is based on simply navigating it? And what would be the key parameters to look out for if not obvious by eye?
Yeah, maybe we can do this one together, Marcus. I think he did a really great job earlier of showing us some of the rainbow streaky thing is a great way to assess if the drift correction is working correctly or if there was any drift in your data. I think that's a good first step. All of our workflows in CODI are these kind of sequential workflows, right? So starting at drift correction and then moving on to the next steps. So if we just kind of follow those linearly, drift correction, is the best way to kind of see if there's visually something wrong with your data or if they needs to be corrected. So that rainbow streaky view is a really good way of doing it. That's just one way to visualize your data, and I think CODI is really great at allowing a user to really display any meaningful parameter that they want to as a color code on your image. So you can color code those localizations for any specific parameter that is relevant to you. So when we're setting those filters that Marcus was showing us earlier and that I was speaking about the overlapping fluorophores, you can color the localizations to give you information about the sigma, so again, that's the size, or the photon count, how bright they are. And you can use those to then set those filters, right? So you can understand your data by changing the color of the localization to, for example, see if you were expecting to see in the example that I had earlier in the presentation, a kind of ring-like structure. If you're only expecting to have some on the outside, but you see some localizations on the inside, it's probably from those kind of double localizations, and you can use those tools that I just mentioned to visualize maybe the photon count or the size, and then set your filters to filter those out specifically.
And then, of course, you have all the graphs and the numbers, and we saw that each tool is very interactive, and so when I click on the graph, it will take me to an example of that particular we were looking at EVs earlier, so I click on the graph and it took me to that particular EV that maybe had was very positive in one of the channels. Similarly, if you see an EV that looks either interesting or suspicious, then you can click on that and it will take you to the same point on the graph, and you can start to use those numbers to see whether it supports your hypothesis, see whether it's something you didn't expect, and then loop back and compare the different conditions to work out why what you're seeing is what you're seeing.
All right, great. So we have literally 1 minute left, so let's try to sneak in one more question. Can you provide an example on how someone might use nearest neighbor analysis?
Ooh, yeah, that's a really interesting one, and stay tuned for more on that one. The example that I was giving earlier about T cells, right, where they were looking at PDL and maybe a different capture antibody, Nearest neighbor analysis is really, really interesting to be able to get the distance, obviously, between clusters that are adjacent, but it can also give you an indication of the homogeneity or the heterogeneity of the distribution of those clusters. So I don't want to lose everybody here, but basically it can tell you, are those clusters homogeneously distributed by looking at the nearest neighbor, the closest nearest neighbor, or the second nearest neighbor, or the third nearest neighbor. Or are they heterogeneously distributed, meaning are they kind of in packets in different places, right? So nearest neighbor analysis is really, really interesting. And again, stay tuned for more on that.
All right, so it seems if we are out of time, if we were unable to get to your questions, we will do our absolute best to contact you and answer them individually. But once again, thank you for the presentation, Corey and Marcus, and thank you to all the attendees for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out the survey to provide us feedback on your experience today. And also, if you're in need of being contacted directly by ONI for more information on our offerings. Once again, thank you and enjoy the rest of your day.
Thanks, Carolyn.
Bye.